The first step of DNA replication takes place at the origin of replication. There are thousands of different origins of replication in eukaryotic cells. Helicase attaches to the DNA at the origin of replication and begins to untwist the double helix, leaving two separate strands. This separation takes place at the replication fork.
After the separation of the DNA strands, single-strand binding proteins bind to the separated DNA strands to stop them repairing, but due to this occurring, the DNA ahead of the fork twists tighter and causes a strain. The topoisomerase breaks, swivels, and rejoins the DNA ahead of the fork in order to relieve the stress caused by the strain.
DNA polymerases usually need a primer and a DNA template strand. The RNA primer is needed to match up nucleotides with their matching pairs. To begin this, the RNA primer first uses the enzyme primase to synthesize the chain. As the DNA polymerase III moves along the unpaired DNA, it adds new nucleotides. As each monomer joins the growing end of the DNA strand, two phosphate groups are lost as a molecule of pyrophosphate.
The parent strands are now separated and are templates for duplication. We use a piece of RNA called primer made by primase to start the process. Then the enzyme that replicates the DNA will synthesize the rest of the complementary bases.
In the lagging strand, since DNA polymerases can only move in a 5 to 3 direction and the lagging strand is in a 3 to 5 direction, the lagging strand sequence of matching nucleotides is different than the leading strand. The primase has to keep adding primer discontinuously and in a series of fragments, called Okazaki fragments. The primase helps the DNA polymerase start the complementary strand just like the leading strand. Sometimes, the DNA polymerase has to go backwards to pair the lagging strand with its DNA complementary strand.
The primase attaches, leaving behind an RNA polymer, and then detaches. The DNA polymerase III then copies the corresponding nucleotides up to the next RNA polymer. The DNA polymerase III detaches and the DNA polymerase I prepares to replace the RNA with the DNA. After the RNA is replaced, a DNA ligase forms, bonding the new DNA with the older fragment. The primase begins to synthesize the RNA primer for the Okazaki fragment. The process of adding DNA to the lagging strand will keep continuing in the same fashion.
The DNA replication process shown previously had DNA while it was still in a simplified form, just in a straight line. In reality, DNA would most likely be in a more complex and sophisticated form, having curves and twists and turns, rather than being in a straight line like it was shown. It shows the processes that occur while DNA synthesis is underway in DNA in a more realistic form.